Two more wonderful guests today. Both are returning. Dr. Jessica Rose will be here in a few minutes. You can follow her on Jessica Loves MJK on X or jessicasuniverse.com. Her Substack is Jessica5B, small b 3. Of course, Jessica has a degree in applied mathematics, immunology, computational biology, molecular biology, biochemistry. She's going to bring us up to date, as is Clare Craig. They're both going to be talking about, amongst other things, self-replicating mRNA vaccines. Dr. Craig is a diagnostic pathologist, author and co-chair of Health Advocacy and Recovery Team. She's been with us before, and after 15 years in the NHS, she became been a pathologist. Training was at Oxford. She has lots of information, and the book is expired. We're going to talk about that and more after this. Our laws as it pertains to substances are draconian and bizarre. The psychopaths start this way. He was an alcoholic because of social media and pornography, PTSD, love addiction, fentanyl and heroin. Ridiculous. I'm a doctor, for Christ's sake. Where the hell do you think I learned that? I'm just saying, you go to treatment before you kill people. I am a clinician. I observe things about these chemicals. Let's just deal with what's real. We used to get these calls on Loveline all the time. Educate adolescents and to prevent and to treat. If you have trouble you can't stop and you want help stop it, I can help. I got a lot to say. I got a lot more to say. Joint and muscle pains are exhausting and frustrating, but I've got a over-the-counter medication I want to introduce you to that provides great relief using the power of, check it out, chili peppers. Capsidin is made with a proprietary formula that contains no non-steroidal anti-inflammatory agents, no opioids, no anesthetic or steroid, nothing, no chance for addiction, no side effects, no chance it's going to interact with other medication you might be taking. Capsidin contains capsaicin, which is the substance in chili peppers that burns your tongue. That gives you that burny feeling. And of course, I've recommended capsaicin creams to patients over the years, but other capsaicin creams burn your skin. That's what makes capsidin so unique. In clinical trials, capsidin has actually been demonstrated not to burn.
I've been using capsidin to relieve my pain in my hands and my wrist from carpal tunnel syndrome and arthritis. The results have been amazing. I use it every day during my show, and I highly recommend it.
Get the pain relief you need from various sources, even backache, sprains, bruises even. Order now at capsidin.com/Drew to get a 15% discount plus free shipping. That is CAPSADYN, capsidin.com/DREW. As I said, the great Jessica Rose will be here in a few minutes, but now we're going to bring Clare Craig in here. Her X handle is at Clare, C-L-A-R-E, Craig, C-R-A-I-G, Path, Clare Craig Path. And also HART.org is the UK's health advisory and recovery team. As I mentioned, her book is expired, COVID, The Untold Story. And she can be found, let's see, where else can we get to Dr. Craig? Well, obviously she has been on, I'm blanking on the nurse's YouTube channel, Help Me Caleb, that has been so great throughout COVID, particularly in the post-COVID era. Dr. Clare Craig, thank you, and welcome to the program. Dr. Campbell, right, I saw you recently on Dr. Campbell's YouTube channel, and I was enraptured with your information and update, and now we got to talk about amplifying RNA vaccines as well. Welcome back.
Thanks very much for having me back on.
So I guess let's start with the book. What can we expect to learn from reading Expired?
So the book Expired is really about the story of 2020. You know, we were told all sorts of things that were not based in fact, and it's it tells a story about where those sort of myths arose. And I dug into the history of it, and it was quite fascinating. I really enjoyed writing it around sort of the myth of the droplet spread, which was so fundamental to all of the beliefs and all of the policies that came out of that. And it's a book that sort of takes it apart scientifically and shows that lockdowns couldn't have worked. You know, we're always hearing that they didn't work, but actually they couldn't have worked. And they couldn't have worked because the virus is spread through aerosols, which can spread long distance through the air. And that's why the time between any kind of new variant and the spike of a wave is the same time. And it's why you had spikes rising in rural areas after lockdown, because these waves are not in human control. They can never be in human control. And it was absolutely insane to pretend they could be. And so, yeah, that's what that book really focuses on. It's really about the virus and about, the kind of exaggeration of the deaths around the virus and about how the virus spread.
And I remember last time we spoke, you had sort of embedded that history in the early days of being contaminated and smells, and how we finally got over that, the idea that these were transmitted through the air in smells, and now we've got something that is transmitted through the air, and we can't get that back in our thinking.
That's exactly the kind of that's the hypothesis in the book, is this problem that we have, that when science gets politicized, which it really was back 115 years ago when people were arguing about germ theory and miasma theory, people get really entrenched in their views. And when they're entrenched, they lose the nuance. They sort of adopt beliefs that aren't quite evidenced, and you have this thing fighting it out. And then, of course, the germ theorists won, but when they won, they brought some of these unevidenced myths with them, and we're still living with the repercussions of it today.
This country, I don't know about the UK, but this country has a long history of doing things like this. People don't understand the opioid crisis is exactly the same story. We developed morphine sulfate and the hypodermic needle. Then physicians addicted millions of people to opiates. And then the Harrison Narcotic Act came out here that literally jailed 20,000 physicians as the source of that epidemic of opiates. For the next 40 years, doctors did not use opiates except under the most extreme circumstances. By the time we hit the 70s, we have people living for decades with cancer and cancer pain that we would not give them pain medicines to. Now a pain medicine group rose up and said, oh my God, pain is what the patient says it is. Pain is the fifth vital sign. You are opiophobic, and the answer has always been there in the poppy flower, and we need to use as much as the patients say. And of course, my patients took full advantage of that, and those same doctors killed my patients by the hundreds. So there we go. Having made it a legal issue, a political issue, not a medical issue, you get repercussions going both directions. And the interesting thing to me about that opioid story is Deborah Birx used the same playbook as the opioid pain medicine group did to bring upon the opioid pandemic here in the United States, which was she went from state to state and got the regulatory agencies, the political powers that be, the state medical societies to sign on, and that was that, and then it was on.
Right, and one of the other overlaps, of course, with the whole opioid story is the oxycodone and is it? Oh, I've got the names. Escape me. It's Purdue, isn't it? The company,
the Sackler family, the Sackler company,
right?
Yeah.
And they had this whole idea about, well, how are we going to sell more of this stuff? We're going to tell people that drug failure is breakthrough pain, and you need more of the drug. So when you have failure, you have more of the drug. And of course, that's exactly what was adopted with the COVID vaccines as well. That, the vaccines failed. What do we need with failure? More of the drug, because we have breakthrough infection.
Isn't it weird? It's so odd how you could put it like a ledger together and document topic by topic how the exact same thing went on in this pandemic. And we need to have like a dynamic explanation and a name frame for it, that if it shows up again, people can go, oh, it's that. Because we've now come to understand this notion of a mass formation, which is sort of how the populace reacts to these hysterical sweeps, these things hysterias that sweep across the land. 20% sign up, 70% just keep their head down, want to be left alone, and 10% go, hey, this is bullshit. Now, but there needs to be a similar way of understanding. That's your next book, Claire. I want you to write the next book about these phenomenon and what we should call them. And please choose a pejorative name. It shouldn't be something lovely. So can you bring me up to date? Go ahead.
Well, no, I was just going to comment on that as well. And I think you're right. And I think one of the things that the kind of that I've observed over the last few years is this sort of invention of vocabulary that gets used a lot by people in power. So there was the invention of some vocabulary like the term pre-bunking. So pre-bunking is this idea that you can immunize a thought. So you can you can take it something that you're worried that people are going to start getting concerned about, and that's going to cause you harm as a powerful person. And you start sharing that, but you muddy the well. You add to it something that's untrue. You then can disprove the untrue bit, and then the truth gets discarded along with that. And then when people meet it again, and the truth, they think, oh no, I've already looked into that. It's not true. So they gave this a term, right? And what we haven't done is give things terms for the sort of opposite effect and the problems that have been caused. And I think you're right. If we had the right vocabulary, it would really help. And one of the examples of having the right vocabulary is the word infection. Right, the word infection gets used as if we all have the same understanding of what that means, and actually it's used to mean lots of different things. So some people will say that you have a COVID infection because you've breathed contaminated air, and that's enough to get a positive test. And so they'll say you've got an asymptomatic infection because you're not symptomatic. I think, well, is that really what we mean by an infection? Isn't that just breathing air? And we all do that all the time. And then people could have the virus enter a cell and their immune system deals with it, so they're still asymptomatic. And why are we calling that an infection? You can't become infectious to anyone else in that scenario, when surely the word infection should be reserved for somebody who could infect somebody else, because that's what the word infection, that's the reaction you get from the public if you've got an infection, is, oh, I might catch something from you. So we shouldn't be using that word to describe things that are not infectious.
Yep, I agree. And certainly infected and infection are different words. Also, I saw on X today, somebody put a headline up. Oh my God, the NHS is reporting that people are vomiting from norovirus in the UK. They are vomiting from the norovirus. And I thought, oh my God. So what is the implication that we should now shut down society because somebody might throw up? Or is it, and this got a little more fundamental to me, that people are unwilling to accept the fact that they are biological agents and as such get sick, infectious and otherwise? It feels like the very nature of who we are as biological beings is being discarded or at least resisted.
Yeah, and I can't help thinking that a little part of that is that the baby boomers have got to the stage where they're worried about their own mortality. Every stage of their life, the world turned itself upside down for their benefit. They were teenagers and they had the sex revolution and they, at every stage. And here they are approaching mortality, and the society is being turned upside down while they're doing it.
Interesting. Oh, it's your next book. Let's put that in the ledger book. But so before we get into the replicating mRNA vaccines, do you have any updates for us on vaccines in general?
Right. So, well, you mean the COVID vaccines? Are we talking bigger than that? I mean, I know you're always in the zone in these topics. So whatever is on your mind and whatever you need to update people about. So the thing that's concerning me the most at the moment is that there seems to be, every now and again, like some messaging gets through and you're allowed it on YouTube and people start sharing it, and it even maybe gets into some media properly. And the message that's being shared a lot at the moment, and it's an important message, I'm not saying it doesn't exist, but there's this message going through again and again about the DNA in the jabs, right? That message is really penetrating. And I just wonder why that is. And I can't help thinking that it might be that's the kind of issue you can say you've solved and then try and resurrect the mRNA platform's reputation and say, well, we've corrected the COVID problem and we're going to keep this platform. And I think there's a lot of other problems with the mRNA platform that have not been heard and that are still being censored because they're problems that are much harder to fix. I mean, this isn't necessarily an easy one to fix, but, it's sort of, you could persuade people you'd fix that one. And so, what I've seen in the big picture, if you just look at what happened when those vaccines rolled out, we had, from spring 2021, a massive rise in the working age population, people who couldn't work because they were long-term sick. And that kept rising up until 2023. It happened in the USA at the same time as in the UK. And we had hospitals suddenly being overwhelmed when they had not been beforehand. And they were being overwhelmed in places like Australia that hadn't really had COVID, but they had also had the vaccines at the same time. And then we saw this big uptick in cardiac problems. So we had This big rise in ambulance calls for cardiac arrests, which has not come back to normal, a big rise in people turning up at A&E departments with cardiac problems, and a rise in excess deaths, which were cardiac deaths. So the biggest problem we've had from these jabs has been a cardiac injury and mortality. And that message hasn't got through. Whereas the problem we've got with the DNA and the jabs, it may well be that these are a cancer risk. And I think the fear of cancer and the fear of dying and the large number of people who have taken these things mean that gets traction because people think it might have happened to them. Whereas, some of the other problems about injuries, they're not really about the person. The message we're trying to get through to people is most drugs, when they're harmful, don't harm all the patients. It's always a minority that gets injured. It's not every patient that's injured. And so the message we need to get through to people is stop thinking about what you've done to yourself, because it's not helping the situation, because you're sticking your fingers in your ears and you're getting scared and you're refusing to listen. Think about what this has done to other people. If we can please focus on that, then we might actually get somewhere. And I think the cancer story, because it's so emotive, is people are kind of hanging on to that and worrying about that, when actually, although I think it might well be real, it's not got the same volume as the cardiac problem, which is big.
And so what you get in this country is tons of pushback on that observation as having been caused by COVID virus itself. So the question becomes, how do we tease out what's COVID? I've been asking this question since 2020, which is what's COVID, what's vaccine plus COVID? Right.
It's not that hard to do, because this was a sort of little miracle that happened, whereby huge swathes of the global population did not have a COVID problem until the vaccines rolled out. And you can use that as a control group, because the timing of any problem should be different for Europe and the USA, which had COVID. Well, not all of the USA, actually. You could probably divvy the USA up into regions that did and didn't have much in 2020. But, broadly, there were regions that had it in 2020, and there are regions that didn't really have a problem until 2022. There's a 2-year lag. So if you're not seeing a 2-year lag in the side effects of these, in the problems that we're discussing, then it's not COVID. It's something else that everybody had in common, which is the vaccines. And more broadly, when you've got a respiratory virus infection, the respiratory virus replicates in the mucosal epithelial cells lining the respiratory tract. And those cells, become little virus factories, and they replicate and they die. And that makes a bit of a sore area, which causes you to have a cough. But broadly, it all happens on the lining of your lung. And you can sometimes detect RNA in the blood in people who get sick. But even Fauci wrote a paper in January of this year where he's saying, well, that, when we detect RNA in the blood, often it is just that. It's just debris from the virus. It's not whole virus particles in the blood. And so the number of people that actually had circulating virus, they would have been really super sick, and the tiny, tiny number. Now, the number of people who've been injected with a product that makes cells all over the body turn into spike protein factories, and then you get circulating spike, that number is enormous. And of course, spike is a pathological protein. And it was, these vaccines had the entire spike sequence. They have 1,273 amino acid protein building blocks. They switched out 2 only. And AstraZeneca didn't switch out any of them. So the entire most pathological part of the virus was being made by cells in the body and was circulating in the blood. So yeah, that's what caused the problems. I don't want to, I understand you being a scientist about it completely. That's how we should approach this. But actually, when you go back to first principles, it's blindingly obvious it was the vaccines, not the virus.
And then the mechanism for the myocardial, or again, what do you imagine the pathophysiology is at the cardiac level that's creating this spike in cardiac events? I hear varying different theories about it. I'll hear yours. I won't adulterate it.
Right. So I think I have a variety of theories. I'm keeping a lot of theories going because I think we don't know. There's, the evidence is still accumulating, and that's fine. And so the big question is, how much of this is from inflammation of the heart. So the myocarditis story, is bigger than what's being presented by the regulators. And how much of it is just barn door coronary artery disease causing infarctions in people? And how much of it is potentially a bit of both, actually. So if we take the myocarditis story first, we know that there were people who had myocarditis and got very sick at the time and had to be in hospital. Mm-hmm. But what we also know is that when 3 different studies followed people up prospectively after their 3rd doses and measured what happened to their heart, having just been given a dose, they had 3% of them had a huge rise in the heart enzyme troponin in their blood, which showed that they had heart damage. So 3% is an enormous figure when you're vaccinating a huge number of healthy people. So we know that heart damage was way more than what turned up in hospital. And that what happens when you damage your heart, the cells can't repair themselves. They're gone forever and you get a scar. And of course, the heart has a very delicate electrical system. So any kind of scarring could mean that you short circuit in the future. And so there is a risk of cardiac arrest forevermore for people who've had myocarditis. And we don't really have a handle on how many people that is and how much of a risk that is. But you would just have sudden cardiac death with no other thing going on. Now, of course, when you have a sudden cardiac death, doctors generally assume it's a heart attack. And so, especially if you're of an age or if you've got other risk factors, they'll say, well, that was just barn door cardiovascular disease that we see, fairly frequently. And so unless you're doing postmortems and actually really looking into it, those are going to be diagnosed as if they are atherosclerotic, narrowing of the coronary arteries, cutting off the blood supply to the heart, damaging the heart, and then death. And so trying to tease those apart is really impossible without proper investigation, which hasn't been happening really anywhere in the world. But we do have evidence that these vaccines cause huge inflammation because of all the circulating spike. There's a lot of inflammation going on in the vascular system. And when you've got inflammation, that is a risk factor for atherosclerosis and narrowing of your heart arteries. So there's a mechanism there for why you would have an acceleration in atherosclerosis. And then going back to the inflammation in the heart, we don't exactly know how the vaccines were causing that inflammation. So the kind of the common myocarditis, well, not common, it was always rare, but the myocarditis that we had before these vaccines would be post-viral, and you'd have a thing called molecular mimicry, where one of the molecules on the virus looks enough like a molecule in the heart that the immune system gets confused and attacks your heart. Right. So that could have happened. That's potential. The spike has a lot of homology, so a lot of similarity to human protein. So that's a possibility. We've seen in postmortems that the spike protein itself was in the heart muscle. So if you've got this product going in and attacking heart cells directly and expressing a foreign protein, then the immune system is doing its job getting rid of that foreign protein. It's a terrible idea to give that to people, but the damage to the heart then is just part of that process of getting rid of a cell expressing a foreign protein. But the other thing to understand is that the mechanism, this kind of mRNA, but also potentially the lipid nanoparticles, has been causing problems beyond the COVID vaccines. So Moderna had a trial with an EBV mRNA where there was a myocarditis problem. And then also there's been HIV mRNA where they're sort of saying, well, you're not allowed to be in the trial if you've had a history of myocarditis. Well, why is that? You're just going to keep gambling with people's hearts like this.
Yeah, I do believe that the spike generally causes an endotheliitis. And so it makes sense to me that both COVID and the vaccine, and the two together, maybe the worst, would cause coronary disease. But I don't see where the virus itself is infecting the heart, and yet we do see spike from the vaccine, right? Would that be a reasonable way to frame that?
Yeah. Absolutely. And what you said about trying to blame the virus, that's exactly what happened when in 2009, 2010, with the pandemics vaccine that caused narcolepsy, they blamed the virus. It's really obvious to try to blame the virus. But if you look at myocarditis rates overall, you say, well, what happened? When was the surge in myocarditis? Nothing happened in 2020. 2020, there was still myocarditis. And some of it may even have been caused by COVID, but it was at the same rates as any other year. So if it was being caused by COVID, then it was replacing other viruses that would have caused it otherwise, right? So nothing happens in 2020.
Yeah.
Spring 2021, massive surge in myocarditis and pericarditis. Yeah. And I think some of that surge is young people who, when they had the vaccine, nothing much happened. And then they're exposed to COVID after they're vaccinated, and their immune system's like, oh, I know this one. And then it attacks the heart.
Oh, that's interesting. I've not heard that theory yet.
Not the unvaccinated.
That's interesting. And by the way, no pericarditis from COVID that I'm aware of. And I remember when I had COVID, I've told this very many times, I had my fever like 103, and I was climbing stairs and my pulse was 60. I went, I think this thing is affecting the heart. But that was COVID itself. But let's quickly, we're going to take a break in a minute, but give us a little sketch on these the self-replicating mRNA vaccines. That just sounds odd and spooky to me. What's going on there?
Right. So, I mean, they seem to just have free rein to do any kind of crazy experiment at the moment. And the idea, the idea that these vaccines are meant to be protecting us from a virus. So you say, well, what is a virus? Virus is an encapsulated code that can replicate. That's what a virus is. And now we've got so-called vaccines, which are encapsulated code that can replicate. You're like, well, at what point are you giving people a vaccine? At what point are you just giving them a man-made disease? I just think it's a really crazy thought to want to do this. And the mRNA that they used in the first place, we don't know even now how long it takes to degrade. We don't know what happens to the synthetic nucleotides that were the building blocks of that RNA sequence, whether the cells carry on using it and recycling it and making proteins last longer than they should. We don't know. And yet here they are saying, well, now we're going to have one that can replicate itself. So I think, we should be pulling back massively from this whole platform, not experimenting further with crazier ideas. I want
to get into this further because I love that frame. So I want to say it again so people understand it, that a virus is a code, DNA or RNA code, surrounded typically by a protein sheath or shell that allows it to get in the cells. The mRNA vaccines are a protein RNA code that is encapsulated in this case, a lipid nanoparticle that allows it to go inside the cell and take over the machinery, which is what a virus does. That they are the same is a fascinating construct, and I want to talk about it in the context of the ethics of doing so, of creating these things, now that it's replicating, it fully is a viral particle, essentially. I mean, the whole idea of being viral is that it replicates. It is a viral particle. And why we don't apply the same standards to this as, say, we do to CRISPR and monkeying with DNA. Why should it? To me, it should be almost identically the same. But let's take a little break. We'll talk about that. We have Jessica Rose in about 15 minutes. Dr. Clare Craig is here. You can follow her on Clare Craig Path and HART.org, the UK's Health Advisory Recovery Team. And do get the book. The book is expired. There it is. The COVID, the Untold Story. Back in a couple minutes. The Wellness Company knows that taking charge of your family's healthcare is a top priority, and being rationally ready, and who knows what the future will hold for us. Now TWC has a service to cover your family's medical needs, including and especially prevention. For just $100 a month, the One Wellness Elite membership includes 2 free medical-grade nutraceuticals per month, free prescriptions for over 800 of the most common medications, access to concierge telemedicine, available at a moment's notice, and a 15% discount on all supplements and the emergency kits. 15% off the emergency kits. That's quite a saving. So if you're spending $100 or more on supplements and meds every month, this plan will already save you money. If you sign up for a year, you'll save $200. And when you use the link, drdrew.com/TWC, you'll get 10% off the first payment to the One Wellness Elite membership. Check out One Wellness at drdrew.com/TWC and get 10% off your first payment. drdrew.com/TWC. It's all there. I want to teach you about pharmacogenomic testing. This is changing the way we approach healthcare by analyzing a patient's genetic profile Pharmacogenomics helps physicians personalize drug treatments for more effective results. First, it enhances medication efficacy by predicting how a patient will respond to a specific drug, reducing the trial and error process, especially in mental health care. For example, it can guide the choice of various medications to minimize side effects and improve outcomes. In the long run, pharmacogenomic testing leads to cost savings. It shortens the time to find the right treatment and avoids expensive side effects. It also supports more personalized care, helping physicians make better decisions and ensuring that treatments are tailored to each individual's unique genetic makeup. Test simply requires you to swab your cheek, then you send in the sample to our lab. Discover the power of personalized medicine with pharmacogenomic testing. Tailor your treatments based on your unique genetic makeup. Reduce trial and error, avoid side effects, find the most effective medication for you. Take control of your health today. Go to drdrew.com/checkgenetics. That is drdrew.com/checkgenetics. I'm excited to bring you a new product, a new supplement, Fatty. I take it. I make Susan take it. My whole family takes it. This comes out of, believe it or not, dolphin research. The Navy maintains a fleet of dolphins. And a brilliant veterinarian recognized that these dolphins sometimes developed a syndrome identical to our Alzheimer's disease. Those dolphins were deficient in a particular fatty acid. She replaced the fatty acid, and they didn't get the Alzheimer's. Humans have the same issue, and we are more deficient in this particular fatty acid than ever before. And a simple replacement of this fatty acid called C15 will help us prevent these syndromes. It's published in a recent journal called Metabolites. It's a new nutritional C15, pentadecanoic acid it's called. The deficiency that we are developing for C15 creates something called the cellular fragility syndrome. This is the first nutritional deficiency syndrome to be discovered in 75 years and may be affecting us in many ways, and as many as 1 in 3 of us. This is an important breakthrough. Take advantage of it. Go to fatty15.com/Dr. Drew to receive 15% off a 90-day starter kit subscription, or use code Dr. Drew at checkout for that 15% off, or just go to our website, drdrew.com/fatty15.
That's brilliant. And thank you, Drew. Where's Dr. Drew? Where is he? Dr. Drew. Dr. Drew.
Clare Craig on X, Clare Craig Path, and Jessica Rose will be here in a second. Jessica loves MJK. So before the break, Dr. Craig and I were talking about the ethics of manipulating the genetics of biological entities, and she was pointing out how much this replicating mRNA vaccine is, in fact, by any description, I don't want to say it too strongly, at least approximates a virus. How do we deal with the ethics, and why aren't we applying the same ethics that we do to other genetic manipulations?
Right. So I just taking that little analogy a bit further. The kind of conventional mRNA ones, they enter a cell with the cell actively uptaking them. They don't just sort of drift through like a ghost through the cell wall. The cell uptakes them up. And so in theory, they can't get back out of the cell. But we've seen studies showing that vaccinated people for 4 months at least had little bubbles of their cell membrane with spike attached to it circulating around the blood. Nobody ever looked to see if there was RNA in it, but there were these sort of, pseudoviruses circulating in the blood in the vaccinated people. So if you can get into a cell and you can get out of a cell, that's like, part of the sequence of what a virus could do. What these replicating ones, one of the differences is, as you pointed out, their capsule is lipid rather than protein. What that means is they haven't got anything on the surface that allows antibodies to bind to say this is something foreign that we need to get rid of. So they're actually like kind of stealth viral type things because they've got nothing, the immune system can't attack them until they've already done something in a cell. So that in some ways they're even worse than a virus would be. But yeah, the ethics of it is something that has just been sidelined, and it's been done very intentionally. And there are lots and lots of strict and difficult experiments that have to be done in order to do anything to do with genetic therapies. And those were all bypassed on the basis that we were in an emergency and then we were going to call these vaccines. And it's as if the word vaccine has this kind of, all-powerful sanctification of a drug, when the drug is still a gene therapy. And it should have had to go through all of the hoops that gene therapy drugs go through. And now that they've done that, they seem to think, well, we can carry on down this path and we don't have to, first of all, go back and do the work on the products that billions of people have had, which have not been done. And second of all, just, carry on with these crazy experiments.
So what were the things, I don't know if this may be unfair to sort of ask you off the top of your head, but what were the usual, I don't want to say even experiments, the usual considerations that were bypassed? So
there's sort of fundamental points around checking whether or not a product like that will enter the DNA, what kind of rate at which it will sort of get into the human DNA. And, the sort of thing I was referring to before around how long it lasts and what that happens to the nucleotides after the product's broken down, what it does for fertility and the germline, all of that wasn't done. It wasn't done. And they just said, oh, we didn't do this. And they were quite open about it because they didn't need to lie because the regulators weren't making them do it. And the regulators still haven't made them do it. That's the real scandal. It's not even been done in retrospect. And if I was looking today at something Pfizer said at the time, back in 2021 on their website, when they were talking about how they had a whole team of people who did gene therapy, and they got that team and they just pulled them into the COVID vaccine work because they had all the experience of their mRNA technology and their lipid nanoparticles, because that is what gene therapy is. They were the experts in it.
I wonder if there's another way to back into this topic through the incredible outrage and the response of the what we used to call molecular biological community to the Chinese scientists who used CRISPR to create a set of twins. That was so outrageous. The level, I'd love to see some of the ink that was spilled on the ethical transgressions of that scientist. And again, using our little new ledger theory that we're gonna one day develop, the opposite reaction that was used in this case to excuse and obfuscate the usual considerations.
Yeah. And, of course, the story that I was referring to earlier that's been shared a lot is around the DNA that was in these products, which were contaminant DNA. And that DNA included regions of sequence that tells the surrounding cell machinery to replicate it and make a particular protein. And if that kind of sequence ends up in your DNA near a whole load of different genes, which are growth genes or replication genes or anything like that, that is a cancer risk. And that sequence should never have been there at all. The DNA should never have been contaminating these vials. The regulator wasn't told about this sequence, and the sequence would have been through a software that would have labeled up what was identifiable and recognizable within it. And that software would have identified these sequences. But what was submitted to the regulator had that information edited out. So that's not a sort of accident or an oversight. Somebody has actually said, well, we're not going to tell them that that's in there because it's a bit of sequence that human cells will respond to, and that it's not good for humans to have. That's essentially what was going on there. And I think there's something about the DNA, which is a beautiful thing. And, our DNA does have within it, human DNA, bits of viruses from over the ages that have sort of, changed what we are and who we are. But the idea that humans are now interfering with what is a sort of divine code, if you like, that there are reasons that we have very, very strict rules on this, because you can't undo messing around with your DNA, and you certainly can't undo messing around with DNA that might end up in the, eggs and sperm and end up in the next generation.
Dr. Craig, I can either let you go or keep you as we bring Jessica Rose in here to be a part of this conversation. I want to interview Jessica a little bit. Do you have time to stay with us, or would you rather kind of wrap it up? We'll get you on You stay? Okay. So Dr. Craig will stay with us. Again, you can follow her on Clare Craig Path on X. Now, Jessica Rose. Jessica loves MJK. Jessica's at universe.com. Jessica 5b3 is the Substack. And Jessica, you heard a little bit of the conversation we were just having. Let me start with asking you the question I was sort of asking Dr. Craig, which was why we didn't apply the same bioethical considerations to these products, in particular this self-replicating vaccine, which essentially approximates a virus in and of itself. Why we don't use the same bioethical considerations that we do, say, with CRISPR technologies, and the outrage that we expressed when a Chinese scientist used CRISPR. We went nuts about that, and yet here we are doing something awfully similar. What are your thoughts?
Because they're calling them vaccines and not what they actually are. So yeah, really nice to hear everything that Claire said. If I may, before I explain exactly what the self-amplifying lipid nanoparticle technology is and why it's more horrifying than the previous version, the modified mRNA LNP tech, I'm really happy to hear that the DNA story is prolific, because from my point of view, it's not prolific enough. It's so important for all the reasons that you brought up about the regulators not doing anything about this residual DNA, which is far above EMA limits, because they're claiming, first of all, they claimed that it wasn't there. Then they checked and they confirmed that it was. I'm talking about Health Canada, the FDA, and the EMA. And now they're saying that it's not a problem. This SV40 promoter enhancer, for example, which is a nuclear localization sequence and a known gene therapy tool, is very bioactive. And they're claiming that it has no functional role. We have FOIA emails that verify this. It's horrific. So the whole story implicates cancer in a nutshell. I mean, residual DNA in products that aren't supposed to contain residual DNA, and certainly not at the levels that we're finding. This is horrific. I mean, it has all sorts of implications for the genome.
You know what, Jessica, I think we all three agree on this, but I'll let Craig, Dr. Craig, address this. I think she was saying that she's worried it will become a false flag, that it's something they could solve and then say, see, everything's fine. Am I getting that right, Dr. Craig?
Yeah, but the thing about it, if I may, is that this is all, I'm going to throw in a pun here, this is all packaged together with the lipid nanoparticle technology, which also is implicated in this new self-amplifying product. So the whole thing is going to go down. I don't think that they're going to just say, okay, yes, we solved the residual DNA problem. I don't think they can, because of why it actually got in there, which is because of RNA DNA hybrid formation. They have been trying to mediate this problem since 2021. We also have a document that proves that. So they've been trying. Moderna's done a better job, according to Kevin McKernan, but I would push back on that. I think that this is the doorway into removing this entire platform from the market. So yeah. And can I comment about the cardiac thing as well? Because hold on 1 quick second. I'll get Dr. Craig's response to the first thing, and then we'll go to cardiac. Sorry.
Go ahead.
I don't think that Jess is wrong. And, the point you're saying there about it, that this is DNA that's actually in a lipid nanoparticle, which will deliver it into the cell with a nuclear localization signal, which drags it into the nucleus. There's all sorts of aspects of it which are really not great. And as she said, they're not very good at getting rid of it yet. My worry is that if this becomes a story, the fact that all of these cardiac deaths have happened will just be forgotten about. And that both things need to be talked about. They're both. There's a sort of the thing that has happened that has killed people, and there's the sort of ongoing risk together. And so, I don't think one story should be dominating over the other. And the cardiac story, I don't feel I guess neither of them have completely hit home, have they? But I'm just hoping that cardiac story doesn't get buried.
I have good news
Jessica.
on the subject. Just today, I reposted a little Senate hearing with Alex Antic, who's a senator in Australia, asking one, I think his first name is John Skerritt, yes, who is a professor, I'm not sure what he's a professor of, about the death of a 7-year-old and a 9-year-old Australian children due to cardiac arrest in temporal proximity to getting the COVID jabs. And the guy said, the professor John Skerritt said that it was more common than people think, dying children, prepubescent children dying of cardiac arrest, which is false, and that that there's no linked signal in terms of adverse event reports and pharmacovigilance databases to the COVID shots. Of course, I had to go right away and prove him wrong on both counts. There's data from the TGA that proves there's no reports of cardiac arrest in prepubescent children like that. And in VAERS, there are thousands of reports of cardiac arrest. We're not talking about myocarditis or any other cardiovascular issues. We're talking about cardiac arrest leading to death. There are thousands of them. And just to give you some statistics here, in terms of temporality, 32% of those reports were reported within 24 hours, 38 within 48, and 52% of all the cardiac arrest reports were reported within 7 days. So this is absolutely linked.
I want to put a little finer point on that. If you remember in the Pfizer research, the original study, they eliminated the first 2 weeks. The first 2 weeks didn't exist. Anybody lost in those 2 weeks was just blinded to the study. Interesting, number one. And then number two, last time we were together, Jessica, we were with Dr. Joseph Fryman, who was pointing out, he was describing such a case of an 8-year-old or a 10-year-old who died immediately following the vaccine to the team at FDA, whose response to the time was, oh, well, I guess it must have fallen through the cracks. And by the way, Still no response last I checked.
Yeah, Senator Antic was like very polite, probably more polite than I would have been. And the Claire, you guys both know what I'm going to say here. The response to hearing about the possibility that a therapeutic product that the children didn't need killed them is just like, there's just no emotional response. And they don't even attempt to engage the possibility that there is a causal relationship. And the data really, really demonstrates that there is. And every single person knows by now, they've heard of somebody suffering from the shots or suffered themselves. I mean, this is, it's not a secret anymore. It's preposterous that anybody would sit there and deny the possibility. Let's just put it that way. I'll be.
And then you want to tell us some thoughts on the myocardial mechanism, the mechanism of myocarditis, or something in there. You said you wanted to comment on that.
Yeah, no, I think Claire summed it up beautifully, and I agree with her. I mean, it's the molecular mimicry doesn't just apply to the cardiac tissue. There are peptides in that spike protein for which there are human homologs, and we can have the immune system attacking anywhere. There's even a paper out there that demonstrates that it can attack immune system cells. So this is really bad.
I'm going to ask you to speculate even further. Is it possible that some of that was constructed in the lab, so to speak, to sort of have this as a bioweapon with maximal downstream effect?
Yes. And the reason I'm saying yes emphatically 3 times is because there's no way that the people who designed the spike protein, it was designed and it was codon optimized, didn't run that stuff through the various bioinformatics softwares to predict whether or not there were amyloidogenic peptides, which there are, whether there was a super antigen site, which it's been documented there is, whether or not there were many homologs to human proteins in there.
Jessica, we know what you mean, but you have to explain that to the public at large. What do you mean? All that needs to be deconstructed for people, just simply, if you can. Homologs, codons, those are the words people don't know.
All right, forget about the codon optimization. You guys don't need to know about that for this, but basically, everybody knows that the sequence for the spike, it's a sequence of letters which are encoded by nucleotides. So a peptide is just a shorter sequence of within that spike protein. So if you chop that up into little bits, let's just say, for example, some of those little bits have exact, it's the same sequence
analogs
in human protein, if you're catching my drift. So they're not similar, they're exact. There's like 100% sequence homology. So if you can imagine,
let's back away from that. There's no way, and I shouldn't say no way because genes are funny that way, but that's not something typically that you would see in a corona bat virus.
Well, I don't know if it would be typical. I don't think it would be typical. But it certainly is strange that there do seem to be quite a large number of these. There are at least 2 papers I can think of off the top of my head that are peer-reviewed. They're sitting on PubMed right now, published, that actually map, many of these. And some of them are actually linked to the female reproductive system as well. But I should tell everyone about self-amplifying mRNA before I run out of time.
Okay, hold on. Before you do, I want to make sure hang on. And Dr. Craig, I want to make sure you don't have anything to say about what Jessica she was dropping bombs all over the place there. And I want to make sure, do you have a reaction to any of that?
No, I agree with her. I think that it was man-made. I think there's plenty of evidence that it was man-made. And there's stuff in that spike that's really nasty. And so, the idea that it would be trying to mimic a human, yeah, sure.
Okay. All right, Jessica, keep going.
I'm just, forgive me, I'm looking for the papers off the top of my head, if I can read them to people about the molecular mimicry. Darn it. I do, here they are. Okay. No, I have it here. I won't even think about sharing my screen. That's too complicated. So there's a paper that was published in 2020. It's called "Is Molecular Mimicry the Culprit in Autoimmune Hemolytic Anemia Affecting Patients with COVID-19?" And begs some other questions, but I implore everyone to read this. And there's also potential autoimmunity resulting from molecular mimicry between SARS-CoV-2 spike and human proteins that was published in 2022 in Viruses. So just to, confirm what I'm saying is true, that there are papers that people have written, that document this phenomenon.
A couple of years ago.
Yeah.
All right. mRNA, replicating mRNA, get there.
All right, guys, sit down. You're going to have to. So I just recently came back from a trip to Japan with some colleagues, lawyers, doctors, scientists, because we had the ear of some Japanese parliamentarians on the subject matter of the self-amplifying mRNA LNP technology. And the reason why we went there was because they were about to launch it into the elderly population starting on October 1st. We did reach people. I think that we actually made a difference. They did go ahead with administering these products. Now, this is a COVID SARS product, okay? This has the spike gene inside, and I'll tell you all about that now. So in my opinion, it's completely moot. Nobody needs to be injected with this stuff because everybody's had an experience with it and a challenge a challenge. But the uptake, the good news is the uptake was apparently quite low. So this is very good news from the point of view of people's awareness having improved. However, because of the nature of these products, it kind of makes me wonder if it's not, quote unquote, too late. So let me tell you what this is and how it's different from the modified mRNA. It does utilize the same lipid nanoparticle technology. The only thing that's different in the formulation that has 4 fats is the cationic lipid. So it's still an ionizable cationic lipid, which is the only thing you guys need to know is that it's highly toxic, and those toxicity issues haven't been resolved. So all the compendial standard issues and the good manufacturing practice issues are probably going to carry right over with these new products. We know how careful they were with formulating and manufacturing these modified mRNA products. We can see because they have residual DNA in them. So we can assume, I'm speculating here, but we can assume that we're going to have similar problems, at least from the point of view of the lipid nanoparticles with this new stuff. So now let me scare you. What they've done is they've taken something called an alpha virus. This is a genus of viruses in the family Togaviridae. Don't ask me how I remembered that. But these geniuses have borrowed a wonderful tool that these alpha viruses use to self-replicate their own genetic material called an RNA-dependent RNA polymerase. So, like many things in biology, we humans look at them, we marvel at it, and then we try and mimic it in the lab for like making therapeutic products. I call it mad scientism. So just like plasmids in bacteria are used in biotech, what these people decided to do was to take this alpha virus and this copying machine enzyme, extract out the subgenomic components, which are the bits of the alpha virus that it uses to give it infrastructure. By the way, the virus that they used is called Venezuelan equine encephalitis virus, and that's as scary as
I was
it
going to
sounds.
say the toga viruses is German measles and equine encephalitis. That's what they cause.
That's right. So the actual
and they
virus
use
that
the actual Venezuelan one, yeah, which is the only one we don't have up here in Southern California. We have everything else.
Maybe soon. So yeah, the story develops. So they're using this the construct of this Venezuelan equine encephalitis virus because it carries this gene called RDRP, that's the RNA-dependent RNA polymerase, that allows that virus to optimize its own survival by being able to copy its own genetic material. So like I said, instead of the virus junk and the replication machinery that they take out, they substitute in the spike gene. So that's the stuff that they put into people in Japan. Now, I'll say one more thing and then I'll tell you why I'm worried about this. So on November 11th, the FDA green-lighted the phase 1 trialing of this exact same technology for H5N1 virus. This is this bird flu virus that they're talking, they're using in the same language or they're using in the same sentence with the word pandemic. It's even in, like, the word pandemic is even in the title of the phase 1 study. And what they've done is instead of inserting spike genes, they've inserted the hemagglutinin and the neuraminidase genes from the H5N1. So that in and of itself is kind of scary. And here's what I'd like everyone to do now as a thought experiment, and the reason why I'm concerned. So as part of this phase 1 trial, there's 200 people in it, 18 to 80. The inclusion criteria list was anemic. They only had 3 points. The exclusion criteria was also, really low. Basically, anybody who had a serious adverse reaction to the modified mRNA shots is excluded. And the inclusion criteria, this is just a point of interest to make you think, included people who were able to get pregnant. If you are of childbearing age, you should protect yourself during the trials. So just want to throw that there.
Shocking.
Nowhere in the inclusion or the exclusion criteria is there any mention of how many shots of the modified mRNA stuff they got, which begs questions about counterindications, or whether or not those people may be carrying replicating alpha viruses? Now, we're going into hypothetical land here, but we have to because of something called RNA recombination, which is a real possibility here. So imagine one of these people is infected with an alpha virus of some kind. All the alpha viruses have this RDRP gene that allows them to photocopy their own genetic material. Let's say they don't have many symptoms for some reason. So they get into the trial, they have some cells. Let's say that some cells have active replication going on in them. And this actually brings us back to that excellent point that Claire made about infection, because unless you have a very high viral load due to a lot of viral replication going on in many cells, you're not really having an infection. So let's just say you have a few cells with viral replication going on. Now, because these new self-amplifying RNA guys are encapsulated in lipid nanoparticles, we know that once someone gets injected with this stuff, that it has the potential to traffic everywhere. So let's hypothesize that one of these lipid nanoparticles reaches a cell in the body that has an active replication going on of an alpha virus. What could potentially happen here, and this is hypothetical, but it could happen, is something called RNA recombination. So during the replication cycle, there's something that the virus can do called template switching. In the presence of this foreign RNA that's introduced into the cell via the lipid nanoparticle, which is, very efficient Trojan horse way to introduce foreign genetic material, it can swap out the photocopying paper, let's say, if you want to think about it that way, and form chimeric RNA. Now, there is a possibility if that happens, because it does happen, that you could actually end up with a new virus. Now, whether or not that, there are a lot of things that have to line up in order for this to happen. So everybody don't get too scared yet. But the thing about this is, and I'm going back to what you and Claire were talking about, this has to have been tested. This has to have been done. These questions need to have been answered way before anybody was injected, because we're dealing with gene-based therapies. And you can't go back from this if we mess this up. And you're actually potentially looking at an actual, biohazard in the making. So it also begs the question, why are they talking about pandemics with H5N1 bird flu all of a sudden?
What do you think? I certainly think that's a sign that they're messing with the virus. Same with the monkeypox. Why does, look, we had indigenous dengue fever break out in Southern California from a mosquito in Southern California, the first time in history. The press unconcerned. It was a major, major, major story in my mind. While there was about 6 weeks ago, it was, I believe there were a couple cases. Yeah, big deal. But in the meantime, a single clade 1 of monkeypox shows up off a plane from Denmark or something, and oh my God, is a headline on the front page of every newspaper. Well, the way this is being reported, the distortions and the lack of understanding of what they're even talking about and what is important and what's not important is making me insane. So I will ask you this question that I asked at the beginning: why are we not holding the same ethical standards that we would for CRISPR generally? Why don't we? What happened? Is it hysteria? Is it that just the fact that it's slipped through and being used so widely, they're just kind of leaning into it? What do you surmise? And let's get Craig in here, Dr. Craig in here on that as well. But Jessica, you first.
I think motive has changed. The definitions are changing. The motives have changed. Maybe it has a lot to do with your point about people just not understanding. And I tend to veer toward that because, just to give you an example, self-amplifying phase 1 trial, if you even tried to explain to the 200 participants what they were being injected with, is there any chance that any of them would understand the implications, the potential, devastation to even to the environment? Because if something goes wrong, these alpha viruses, they're shared among many mammalian species. So it's like, we're not just talking about messing up our own, let's say gene pools, I suppose. When you're talking about gene-based therapies, it can translate, pardon the pun, to other.
The point is, it's I think the way to say it is not just the individual's, say, cancer risk downstream, the many other risks. Dr. Craig, are you still with us? And want to get your thoughts on this same topic. Go ahead there.
I am still here, and I can't help thinking it's something around scientists seeing the sort of DNA world as a final frontier, and that there's some sort of, we have to do this because this is progress. And I think they've managed to convince a lot of powerful politicians around them that this is progress. And the reality is that a lot of what they are claiming that these products can do is based around mythology in the first place. So, when Jess is saying, well, people don't need these injections, they never needed these injections. They were never ever needed. And a lot of the sort of stories told about viruses and about pandemics are mythological. They're not based in fact. And they've over time, they become more and more scary. It's incredible how the numbers escalate over time if you look through history. So 1918, at the time, they thought it was about 20 million people who died. And that was probably an exaggeration. And now people like the Wellcome Trust will say it's 150 million. Like, well, what happened there? Because they could count dead bodies back then. They're pretty good at that. So there is this whole fear-based thing that's driving all of it, which is not good for us.
Hysteria, mass formation. It feels like that's been a lot of the story of the last 5 or 6 years. Is there more to get into, Jessica? Have we run the cycle? We covered the topic to your satisfaction?
I think so, yeah. I can't emphasize enough how people should stay well away from any gene-based products. That includes the modified and the self-amplifying stuff. It's even more red flaggy to me. Because the potential, quite frankly, when you start messing with genes, like I said before, there's no predictability here. And we actually could end up screwing ourselves, pardon my language. I know that fear is a huge component, and without it, they wouldn't have gotten away with what they got away with in the first 4 years. And I think that, people absolutely need to be based in knowledge and not fear. But at the same time, man, we have to scream to the rooftops, enough with the gene-based stuff, enough with the gain-of-function research, enough with the chimeric virus crap, enough with building viruses so that we have to make vaccines before that guy gets us. I mean, the whole thing is so absurd. In my opinion, we're brilliant. Our species is so brilliant. And all of the stuff that we could be doing and all the energy and the time and the money could be spent on such amazing stuff. Not this biohacking nonsense that will end up leading to our detriment. That's all I have to say.
Okay. And Claire, any last thoughts?
I'm with you, Jess. It's fear sucks something out of people. We've had enough of it. And as you say, there are we just want people to be looking to the future in a more positive way instead of in this reckless way. Just that there are much more constructive things people could be doing with their lives.
Yeah, I'm just thinking, hearing your beautiful accent and then talking about the destructive effect of fears. I had a historical sort of weird image of the idea of Winston Churchill going, "Shelter in place, shelter! Oh my God, we're doomed." I mean, just look at where history has gone well and where it's not gone well. It's never because of fear. It's not gone well because of fear and panic. And anybody that has addressed horrifically and genuinely fear-provoking circumstances did so with intestinal fortitude, and things went a lot better. Guys, thank you so much for being here, and thank you for all the work, and I hope you'll keep it up. We're going to talk about Claire's book. Jessica, do you want people just to go to your Substack?
Yeah. Oh, by the way, I just wanted to mention my website is down, the Jessica's Universe. I'm having domain issues. They're linking it to something that you don't want to go to. And my Substack is also justr.substack.com. So I have two.
What is it? Say it again. R?
Jessica R, like jessicar.substack.com.
Jessica R. Okay, Jessica R. Got it. Jessica R. And then let's put up Claire's book one more time, Caleb, if you don't mind. Want everyone to read that, and it's a really interesting expired the untold story, COVID the untold story. And it goes back through his history of infections generally and our ideas about them, and how we have been infected by ideas that have affected our response to these sorts of outbreaks. Thank you guys so much. Hope to talk to you again very soon.
Thank you very much.
Thanks for having me.
Of course. Jessica's at Jessica Loves MJK on X, and Claire is Clare Craig Path. Coming up for us, we have Susan's show this afternoon. She has a very special guest. We're not telling you who because we don't want to adulterate or have there be the potential for any claims that the psychic knew who was coming or looked her up or looked them up or something. Andrew Grohl next week with Wilk Wilkinson, Ted Balaker, Courtney Moorhead Balaker as well, Josh Bernstein, Allison Monroe. Didn't we have Laura? I don't know what happened to that. Justine Bateman coming in, and Jeff Dye. Should be interesting once we get into December. And of course, Kelly Victory sitting in for me, interviewing Ed Dowd on December 4th. So keep an eye on all that. Let me quickly glance out at the restream, seeing what you guys are doing. Thank you, Gene Feist, for the comment about the show. When am I going to acknowledge the fact that I played a part in the mess at the ONS? Can somebody tell me what the ONS is? I played a part in the mess at the ONS. Caleb, do you know what the ONS is?
I'm trying to figure that out, but who knows?
I played a part in the mess of the ONS, I guess. I'm going to look it up.
Isn't that great that someone's mad enough at you to come on the show and leave a comment, and you're like, well, I don't even know, what did I do now? What are you mad about? I have no clue.
Yeah, I don't even know what they're talking about. Okay, well, good. I'm getting used to it. Let's see, pandemics, many have stopped seeing their doctor. That's not good. Office of National Statistics. I caused a mess at the Office of National Statistics. Caleb, any theories how that might have happened?
Never heard of that either. That's not on my list of usual complaints that people have about you. No. I'll add it. I'll figure it out.
And I'm also looking at you guys on there's a lot of stuff on the rants I missed here. Give me a second here. I had my cup up here, guys. New mosquitoes are of the Aedes species. They can host dengue, unlike the local Anopheles species is disturbing. Aedes are not naturally home in Nebraska. That is true, and that's why the fact that, of course, where do you think people go with the fact that we have the non-Anopheles Aedes species up here? First thing they do, they go, oh, it's climate change. Okay, that's what did it. That's it. By the way, there is a doctor. We should interview my friend now. He has a whole theory that hold on a second. A whole theory that the Zika virus story was also an hysteria, because it turns out that really never happened. So I need to get to Dr. Emily Barsh about that to see if we can interview the doctor. I'm blanking on his name now. I've got it on here, but it's not showing up. Caleb, anything else on your front?
Nope, just excited. We got Susan's show coming up with a surprise guest later on today, and big list of interesting people coming up next week. Very excited.
Yeah, it's going to be very good. All right, everybody, thank you all for being here. We'll see you at 3 o'clock this afternoon for Susan's show. And I think we're in, let me double check the dates. Hold on here. We're not here on Monday. We're here on Tuesday at 3 o'clock with the Gruel, with Andrew Gruel. See you then. Ask Dr. Drew is produced by Caleb Nation and Susan Pinsky. As a reminder, the discussions here are not a substitute for medical care, diagnosis, or treatment. This show is intended for educational and informational purposes only. I am a licensed physician, but I am not a replacement for your personal doctor, and I am not practicing medicine here. Always remember that our understanding of medicine and science is constantly evolving. Though my opinion is based on the information that is available to me today, some of the contents of this show could be outdated in the future. Be sure to check with trusted resources in case any of the information has been updated since this was published. If you or someone you know is in immediate danger, don't call me, call 911. If you're feeling hopeless or suicidal, call the National Suicide Prevention Lifeline at 800-273-8255. You can find more of my recommended organizations and helpful resources at drdrew.com/help.